Hi, the disease that we wanted to help develop a vaccine for was Middle Eastern Respiratory Syndrome, also known as MERS. Starting off with our abstract right here, some key points would be that MERS is an illness that is caused by the MERS-CoV virus. It is also zoonotic, which means that it's transmitted between humans and animals, specifically the dromedary camels. It's also transmitted between humans through close contact and respiratory droplets. Like any other respiratory virus, MERS is highly transmissible, meaning it is very easy for outbreaks to occur. A viral vector vaccine would be the most helpful way to be able to stop the prevention of infection with the MERS-CoV virus. And here are some reasons why. But first, let's start off with some background knowledge of MERS and let's look at the literature review next. Like previously mentioned, MERS is a zoonotic disease, and the reservoir hosts are the dromedary camels. They're very common in the Middle East, making it very easy for the disease to be spread. Some examples of the interactions that they have with humans are the usage of them for transportation, hauling, and ingestion of their milk and meat. Majority of the cases that are reported are from the Middle East, especially those that are traveling to the Arabian Peninsula. And lastly, a major virulence of MERS-CoV are the spike proteins that are presented, which help the virus to be able to attach themselves to the cells, and this is how they're able to interact with the proteins of the human cells.
Talking about the design of our MERS vaccine. Our MERS vaccine is an adenovirus viral vector vaccine, which means we took a mild form of a virus, adenovirus, and incorporated the genome of our virus into a plasmid and put that into the adenovirus. We chose the adenovirus because it's a common virus that infects humans, but it's very mild, so it's not going to cause severe infection when being used in a vaccine. The means by which the MERS virus infects host cells is through binding of their spike glycoprotein to a host cell receptor called DPP4. To establish immunity, we want to disrupt that interaction. So to design our vaccine, we took a plasmid and recombined our viral spike protein gene into it, and then purified it and cultured it into human cells. Once we had our vaccine, we determined how immunity is established. Once the adenovirus viral vector enters the body, it binds to host cells via TLR receptors. This causes a signaling cascade, which releases cytokines and chemokines, and this gives us our innate immune response. And then we have our adaptive immune response, which is given by the expression of our viral spike protein gene, which has been expressed by the host cell and delivered by our viral vector vaccine. Once this is expressed, we get antigen. The antigen is taken up by antigen-presenting cells like dendritic cells and macrophages. MHC molecules on the surface of antigen-presenting cells present the antigen to T cells. T cells are then activated and they will differentiate into CD4 positive T cells and CD8 positive T cells. We also have antigen binding to B cells, which will activate the B cell, and they differentiate into plasma B cells that produce the antibody. The antibody will then bind to the glycoprotein on the surface of the virus, so it inhibits the interaction between that surface protein and a surface protein on the host cell. And then we also have differentiation into memory B cells, which gives us our immune memory.